He comes from University of Pennsylvania, the famous Translational and Correlative Studies lab at UPENN, who had developed novel immunotherapies for treatment of cancer. She will tell us all about it. Chelsea is a young investigator. She's a biologist. She's been with the specific Translational and Correlative Studies lab at UPENN since 2014, and she's also a post-bac student at UPENN, which means that soon she will be targeting a graduate PhD or MD, PhD most likely degree. I think it's fascinating because it's a new era. So far, all what we knew about, allow me to say that, about cancer therapies was surgery, radiation, chemotherapy. So now we entered what they have done at UPENN is they open a new gate for immunotherapies, which is amazing, actually. But she will tell us all about it. Chelsea, without further ado, Chelsea Bartoszek. Thank you.
Thank you, everyone. Thank you for having me here. So what I'm going to talk to you about today, as the title says, it's a therapy that's driving an immune response. And what makes this so interesting, so cool, is that this is the first therapy that's been FDA approved that is a living drug. So this isn't like anything that's been on the market before, and it is there now. And it is something that's so incredible. I'm grateful that I've had the chance to be a part of it and that I could come and tell other people about it so everyone knows, as Elias said, this new era that we're moving in as far as biomedical-based research. So before I get into what this therapy is, I want everyone to kind of be on the same page as far as immunology, because the core of what it is it's an immunotherapy. So we need to kind of understand what that means. So as far as immunology is concerned, your body has some major defense systems. The first one is very basic, and it's not something that you might consider to be even part of your immune system. Does anyone have any ideas what might fall under the category of barriers? So what are some basic things about your body that keeps things like bacteria and viruses from getting to you? Yeah, your skin is the major one. That's why anytime you have any sort of major surgery or anything that leaves an open wound, you are much more susceptible to catching a variety of things because that is your major barrier. Does anyone have anything else? So another basic one is the mucus in your nose. That's another big one since we breathe in so many things every day. That's something that is preventing that from actually getting into and infecting our bodies. So the next line we go into moves into what's actually generally more considered to be a part of your immune system. So we have cells that fall under this non-specific category. So they're immune cells, and they're fast-acting. They recognize when something bad is in your body, but they can't really identify what it is, and they don't always know what to do with it. But they can get there first, and they can start doing damage control and move on to the next set of cells. So your next set in your immune system are very specific cells. They're very targeted, they know what they're going after, and they're able to kill it, but it's much slower. So if you had to rely solely on that, we'd probably be in trouble. Also very cool about your specific cells is that they're able to create memory. So these cells could remember things that enter your body, and they could be fast acting for the next time if anything similar enters again. So there are two main branches of your immune system. You have your innate immune system and your adaptive immune system. Now your innate immune system are the cells that fall under that non-specific category. They do lots of great work for you as far as getting rid of infections or things that are attacking your body. But they're not specific, and they don't have the same types of techniques that the adaptive system has. One of the other major things that the innate system does, though, is it talks with the adaptive immune system. So it needs to, when it realizes it's in over a cell of your innate immune system, it gets in over its head, but it knows something is bad and needs to get out. So then it could go and start talking to your adaptive immune system and gets that involved. So for the purposes of this therapy, your adaptive immune system is really where it's at and what's important to us. And there are two main cell types that are involved in your adaptive immune system, and those are your B cells and your T cells. So T cells, they come in a few varieties, but there are two main types. You have your cytotoxic T cells, which were formerly called killer T cells, and they're the T cells that once they become aware of something going wrong in your body, they could actually go in and eliminate it. Your helper T cells can't do that, but they're equally as important because they're the ones that's doing all the talking between the cells in your body. They are also able to identify what it is that's infecting you, and they get all the cells that need to be involved and know what's going on. So moving on, the other main cell type of your adaptive immune system are your B cells. Now, when your B cells are exposed to something that's generically referred to as an antigen. So an antigen could really be a variety of things. We're talking, it could be a virus, it could be a bacteria, it could be a chemical that you inhale, and now that molecule's in your body. And it could be cancer too. So anything that is not supposed to be in your body is termed an antigen. So when your B cell sees this antigen, so you see something that's going wrong, it develops into a specialized B cell called a plasma cell. And at that point, once it becomes a plasma cell, it could secrete these antibodies. And antibodies are these Y-shaped molecules, and they are designed to bind to this antigen. And the binding is very interesting because it is very specific. So you see in this little diagram, it's showing antigens of different shapes and colors. But this particular antibody can only bind that one antigen. That's it. So how does that work? Well, it works because your body has over 10 billion different combinations of antibodies. So if something's entering you, there's going to be an antibody that can identify it. And once it does, it's going to grab on, hold on tight, and it's going to neutralize it. So it can't go on to reproduce, and it can't infect you anymore. And then it's going to call in other cells, such as your cytotoxic T cells, to actually eliminate it and officially remove it from your body. So one major thing to note about all cell types, this isn't just your immune system, this is everything, is that they all have these markers on the surface of their cell. Often they're designated with this CD before a number, but they can be named a variety of things. They do a variety of different things as well, but the important thing to know is that they're involved in sending signals, so they're involved in your cell's communication. As far as research is concerned, they are the prime way that we are able to identify these different cells because there's generally some form of uniqueness that we could look at. So this picture shows way too many different ones that we're not really concerned with. But a few of the important ones, if you're looking at a T cell, CD3 is the marker that tells you that you're looking at a T cell. From there, you have two options. It could have this other marker called CD8, which tells you that you're looking at a cytotoxic killer T cell, or it has a CD4, which tells you that you're looking at a helper T cell. As far as B cells concerned, CD19 is where it's at. And I will explain why this marker is so important, but it's sort of the crux of where this CAR-19 therapy came from and what it's all about. So moving into this therapy, I think it's important to see where we've come from and that it's not a therapy that was even worked in the last decade. It's been much more time than that to actually go in to making this thing happen, to bringing this new line of research to the medical field. So one of the first key points that I thought was relevant goes way back to 1891. And this was the first time that a doctor was attempting to harness the power of your immune system to treat cancer. So Dr. Coley is termed the father of immunotherapy. In his early years, he was interested in cancer from the start and got discouraged by the fact that he was doing all he could for his patients, as far as even going as far as amputations, serious treatments that were the best they had at the time, and he didn't think that was good enough because he was losing patients. So around the same time, he became aware of a case of a cancer patient who had cancer and then became infected additionally with a bacteria. And it was a serious infection. The patient eventually recovered from the infection, only to find that their cancer was also gone. So he had to know what was going on here. Like, why would that happen? This cancer that was shown to be untreatable by anything else they had at the time. So he started looking into the cases. And from there, he found 40 different cases that all showed a similar kind of phenomenon, where a cancer patient becomes infected, they recover from the infection only to find that their tumors have shrunk, and some might even be in complete remission. In other words, the cancer was undetectable. So, because this was 1891, he decided to test this. And he took sarcoma patients that had essentially untreatable cancer and infected them with live Streptococcus bacteria. And he kept infecting them till they formed a serious infection, then tried to tackle the infection, and found that, again, his patients' tumors were disappearing, that this was having an effect on their cancer. Now, obviously, there's issues with ever infecting someone with a live virus, so eventually he did switch to using dead or otherwise inhibited bacteria to try to control the situation a little better. But the point was, he realized that this was because this massive infection was causing your immune system to become engaged again. It was waking up your immune system, and that's what was having the effect on cancer. And this line of thought is particularly awesome considering the fact that only about 30 years earlier did they start to accept that bacteria cause disease. Before that, physicians would walk around in dirty smocks and thought that meant they were a really good physician because it showed how many surgeries they had done. So this was an awesome line of thinking that your immune system could be this kind of secret weapon if you only figure out how to use it. So one of the next big discoveries as far as leading to immunotherapies is concerned was in 1956 when the first bone marrow transplant was performed. Dr. Thomas here was the first physician that actually performed this transplant. He had a case of identical twins. One of the twins had leukemia. He gave that twin chemotherapy to wipe out their bone marrow. And in your bone marrow is basically where all your blood cells are developed. So this includes red blood cells and all your white blood cells, so your cells of your immune system. So it wiped all that out. And then he took bone marrow from the healthy twin and put it into the sick twin to cure the leukemia. And this, as a side note, this is also his wife Dorothy is in the picture because she was instrumental in doing this research and figuring out what was going on. And together they are thought of as the parents of bone marrow transplants because they were the first ones that really investigated it. So here, again, we have a first time that we're taking cells from outside of a patient, putting them back in a patient to treat cancer. So then only a couple years later, we had the next success, which was an allogeneic bone marrow transplant. That's different from using identical twins because it's not an exact match. The reason why they first went with twins is because then there wouldn't be a fear of the body rejecting this foreign invader, because the genetics are the same, so the cells should be the same, and they'd recognize them as such. But Dr. Mate here decided that you didn't necessarily need identical donors, because how many people have identical twins hanging around to use if you get sick? So we need something. So everyone else in the room needs something, something more to help them, because they don't have that option. So he did the first bone marrow transplant on unrelated individuals. And actually how it happened was a group of physicists were doing some sort of study. They were all accidentally exposed to lethal levels of radiation. At least one of them died. The rest of them had no hope. So they were like, yeah, transplant my bone marrow. Let's try this. And he did. And it proved successful. Now, the problem with this, and why there is still a focus on trying to find matches between patients, is that the recipient patient, the one that gets the donated bone marrow, could have a chance of developing what's called graft versus host. And that's actually where these healthy cells that are infused into the patient, they recognize the patient's body as foreign and begin attacking everything. And oftentimes ultimately kill the patient. But as time went on, doctors started looking into this, and they realized that patients that got bone marrow transplants from non-identical donors, even if they developed graft versus host, and they ended up surviving it, they had lower rates of their leukemia returning than identical matches. So this was interesting, and what they began terming a graft versus leukemia effect, where essentially what they surmise is that the donor cells, the cells that you're putting into the sick patient, end up taking control of the body. They become the dominant cells because they're the healthy ones, and they're wiping out any remnant cancer because they don't recognize it as their self. So this was a case where, again, your immune system, or a donated immune system, as it was in this case, is acting as a cancer treatment. So then going on to 1987 into the early 90s, we have the first concept of a chimeric antigen receptor T cell is thought up. Up until this point, people had been working on taking T cells and modifying them, working out ways to grow them outside of a body. And it built up until this point where Dr. Ashar engineered the first CAR T cell, what we call a CAR T cell. So that's great. So now we need to actually talk about what is a CAR T cell, what makes it different than a normal one, why is it special, and why is it potentially effective. So the whole thought behind it was that our immune system is great. It does so many things for us, but for a variety of reasons, depending on what we're targeting, it fails. So we want to make a better immune system. We want to create these super soldiers that are able to go in there and do their job at the highest level. So then the question was how to do this, how to make them better. So if you see from this picture, what's labeled as a conventional T cell, so that's a normal T cell that you'd have in your body, it's a fairly complicated interaction system. You have your T cell receptor that, just like the antibodies, it's very specific. So it could only recognize one type of antigen that's infecting you. But it doesn't end there. There's also different connections that need to be able to be made for the T cell to actually recognize the antigen. So it's a multiple step process. So when they were thinking about CAR T cells, they thought that was a little complicated. They wanted to streamline the process to have this thing to go right to the cancer and to attack it. So they decided to make some modifications. And so if you think of what the term chimera means, if you've heard of it before, you've probably heard it in context of like an animal where it's like you have the head of an eagle and a part lion and part snake all thrown in together. So chimera is basically just referring as a mismatch of different things. So what a chimeric antigen receptor is, it's sort of taking the best of both worlds as far as an antibody, which would normally be found on a B cell, attaching that to some components that are normally found on a T cell. And the big deal is that they're able to take that part, which would normally be on a B cell, and engineer it for a specific target. So they want to send it after some certain type of cancer. They can engineer it to look right for that type of cancer. So in this picture, again, what's labeled as a TCR is just like a normal, the normal receptor that would be found in your T cell. This first generation CAR here is what Dr. Ashar, the first guy that came up with CAR T cells, this is what he came up with initially. And basically, it was just, you see the little turquoise things. Those are like replicating sort of like an antibody. And he just attached it to a T cell. So that was all it was. And it was great. He engineered those little turquoise bits to find what he was looking for. So it attacked, it recognized the cancer. But the problem is the T cells had no means of proliferating. They had no means of dividing and actually behaving as normal T cells would. So there was no permanence at all to them. So they weren't very effective. So then the second generation CAR, which is getting into what the University of Pennsylvania developed, they started adding components that are normally found in your normal T cell to it. So normally a T cell will require different signaling that will tell it to divide, for instance. So they added these different signaling molecules that would convey to the T cell that, okay, you can't just attach to this cancer, you also need to take care of yourself, you need to proliferate, you need to get over. And then with the third generation CAR, it was close to the same thing, but they added a secondary signaling molecule just to try to increase these T cells' longevity and permanence in your body so they could keep monitoring for cancer. So, like I said earlier, CD19 is very important, and it was kind of started this whole therapy and what made it a success. But why is that? So one of the great things about CD19 is it is expressed over a wide range of B cells. So like most things, B cells have multiple levels of development. You have like your little baby B cells that pop out of your bone marrow, and then they start maturing, and there's multiple levels. But CD19 is found on all of them. So whatever B cell gets infected by a cancer, it could still find it. And this also goes hand in hand with the fact that a lot of times when a normal cell turns cancerous, its markers do change. So say a cell has something like CD138 on it when it's healthy. It becomes cancerous, and that's gone suddenly. However the cancer is working, it doesn't need that marker anymore. But with B cells, that's not the case. Even when they become infected, that marker is still there. And because of that, it's present on most, if not all, different B cell cancers that are out there. And the final great thing about it is that it's not actually expressed on stem cells from your bone marrow. So ideally, long term, once the therapy has been successful, these B cells could eventually start growing, your body could start growing new healthy B cells because the stem cells are unaffected. And hopefully, T cells will still be around if anything goes wrong with those B cells. Oh, and also it's not affected, it's not on any other tissue in your body. It is literally just B cells. And while not ideal, people can live without B cells with just a little bit of help with some immune boosting techniques. So getting closer to present day. In 1998, we have when Carl June, who is my big boss, the big deal at the University of Pennsylvania, essentially, he was first funded to start doing CAR T-cell research. And then 12 years later was when he was able to open the first clinical trial. So it took 12 years after we knew in the early 90s CAR T-cells had been engineered, but it still took 12 more years of research before he was actually able to open a clinical trial. And with that clinical trial, he was only able to get funding for 3 patients. That was it. But one of the great things that happened from that is that 2 of those 3 patients, and all 3 patients had what's called chronic lymphocytic leukemia, which is a type of long-lasting leukemia that's prevalent in adults, particularly in the United States. And all 3 of them had this disease. They were treated with it, and 2 of the 3 of them went into full remission. And not only did they go into full remission, so their cancer was undetectable within a few weeks after this treatment, up to 8 years later, I could confirm, because I just saw the data, they are still fully clear of their disease. So that was an awesome win, but it's still only 3 patients, and it only worked on 2 of them. So that wasn't even enough to publish a paper, let alone to really get anywhere in the field. Fortunately for Carl June and this area of research, the drug company Novartis saw something in this research. They thought it was going places, despite the small sampling that they had. So they entered into a unique partnership with the University of Pennsylvania. It was industry and academia bonding, which they don't generally like to do. And they worked together, and Carl June got more funding. So he was able to start with more patients. Unfortunately, things still weren't going the best. They knew some things about this therapy, but anytime you're working with human trials, there's bound to be issues because everyone works differently. How this therapy reacts in everyone is going to be different. So they were having setbacks, namely which patients were dying from this therapy, and they didn't quite know why. Because they had these first 3 patients, none of them died, and it was totally successful in 2 of them. So it was unclear what was going on. So the next big deal with this therapy, the reason why it's pretty universally acknowledged, the only reason why this was able to get where it is today is because of one certain patient that was our first pediatric patient to receive the therapy. And her name was Emily Whitehead, she was diagnosed with acute lymphoblastic leukemia at 5 years old. This disease is very prevalent in pediatric and young adult patients. And it's a very aggressive form of leukemia. So while chronic leukemia sufferers will generally live years and years with this disease, acute leukemia people don't really have very long and generally don't have very many options of viable therapy. So she became the first pediatric ALL patient that became enrolled in a CAR-19 trial at the University of Pennsylvania in conjunction with Children's Hospital of Philadelphia. And they were excited on trying this up until the point that she got very sick less than a week after receiving the therapy. And they didn't know why. So what's particularly cool and close to home about this study for me is that they consulted with my lab. This was before my time, but they were like, we need data. We need to see what's going on. So we rushed some data over, and they identified what was causing her sickness. And it was something that they didn't have a term for at the time, but is now termed cytokine release syndrome. So basically what happens when you get this treatment is your body enters a form of shock because you get this treatment, your cancer is being attacked, all these signals are going on all over the place. Cytokines are just your body's way of sending signals out to each other. So it's sending all these signals, and your body can't keep up. So it goes into shock, and your system starts shutting down because it can't handle it. So they identified a specific cytokine that was causing the problem. And the clinical side, her doctors had been monitoring cytokines, but they were focused on cytokines directly related to T cells because they thought T cells are what's being added to her body, so that would be where things would be going wrong. But it was actually a cytokine that was not directly related to T cells. So now they knew what it was, but they had no idea what to do with it still. So I don't know about you, but like, what do you do when you have a question that no one around you can answer? I consult Google. And that's what the doctors did too. They were rushing, trying to find something that would save this girl's life. And they weren't really coming up with anything until Carl June's daughter happened to have juvenile rheumatoid arthritis and had just started a new medication that attacked this particular cytokine. So and it was fortunately a drug that was readily available in the hospital. They got it to her right away, and she recovered fully. And not only did she recover, but at the end of it all, her cancer was gone. And just under 6 years later, she is still in remission. And her family is a big supporter of the therapy, and she's sort of become the poster child of it. So after the success of Emily Whitehead, there was many more pediatric patients, hundreds, that were treated and was showing great success. In their initial trial, we had around 90% remission rates. And so it was great. And the FDA recognized it as such. And in 2014, at the University of Pennsylvania, our CAR-19 therapy, also called CTLO19, was designated a breakthrough therapy. And what this basically means, it's put on the fast track to actually getting approved as a drug through the FDA. So if you know anything about the FDA, fast track is only so fast, but it's still very exciting that just over 3 years later, in August of 2017, the drug Chimeric, which is our version of a chimeric antigen receptor targeting CD19, was approved by the FDA to treat acute leukemia. And then a secondary excitement is in May of 2018, that approval was actually extended and is now in use for treating lymphoma as well. Okay. So I just wanted to briefly kind of go over how these CAR T cells are made and what the whole process is behind it. If you look into CAR therapy at all, one of the big setbacks is it is a pricey therapy, but there's a reason for that. It's very specialized because the first step is we have to get cells from a patient. So there's this process called leukapheresis where patients' white blood cells are separated from their red and collected in a little baggie. This baggie is then goes on to a specialized cell center where the process begins of actually creating these CAR T cells. So what they do is first, this is just a collection of all white blood cells. So that's T cells, B cells, and all cells of your innate immune system could all be mixed up in there. And we only want T cells. So using those markers, like I told you, the CD3 and the CD8s and 4s, these markers that are known identifiers of T cells, they use that technology to isolate and separate the T cells from the rest of the cells. So at this point, we have a nice collection of the patient's T cells, but that's still not CAR cells. So from there, we need to actually add this antigen receptor onto these T cells. So we take a lentiviral vector, which is basically using a viral vector similar to what HIV is. It's inactivated, so it can't replicate at all, but it can still infect. So within this little vector, it's a whole bunch of genes. And importantly is the genes that tell the T cell how to make this receptor. So this virus infects the T cell, inserts these genes into the T cell, and now the T cell knows how to make the receptor. And so now these T cells have become chimeric antigen receptors. So from there, it's basically just about expanding. A lot of these patients are very, very sick, so we get a very small amount of T cells at the start, and we need a lot more if we want the therapy to be successful. So over a course of 9 to 11 days, these cells are just grown up outside of the body. And at that point, the cells are what's called cryopreserved. And it's basically just a fancy way of saying that the cells are frozen down and stored. And the reason that they're not put directly back in the patient is because we need to test this. These labs are they're formed to not infect this product, to not contaminate these cells, but we need to be sure before we're putting this back into a person. So after a bunch of testing is done to confirm that they do have the CAR, so that part, the infection process was successful, that there's no contamination, then the samples are thawed and a certain dosage is put back into the patient. And from there, they take over, which is the awesome and terrifying part of a living drug, because you don't know what it's going to do once it gets in there. When we have an idea, we have hundreds of patients now at this point that gives us a better idea, but it's always still something new to see what's going to happen. The whole process, it's probably about 2 weeks, maybe a little bit more depending on how the testing goes. But yeah, about 2 weeks. And that's another good point, because all of our patients, this isn't a first-line therapy. And what that means is the patients have to be very sick before they qualify. So they have to have tried all other therapies, essentially, and have it not work. So these patients are very, very sick by the time they get to us. So time is of the essence. And that's why a lot of work has gone over the years to perfecting this particular process, not just improving the CARs themselves, but this process to make it better for our patients. So we have CAR 19 on the market now, which is fantastic, but doctors see immunotherapy and CAR therapies, it's the way of the future, essentially, how we're going to be fighting not only cancers, but other infectious diseases. There's lots of HIV trials with not exactly CARs, but similar types of technologies that they're trying. So some different things that they're looking at is trying to use different targets instead of CD19. So CD19, as I discussed, is really great. There's a number of reasons why it works so well, but they're trying to find other things that might work better for different types of cancers. Even B cells, you would think this is a treatment that targets CD19. It's on all your B cells, so it should easily find and attack all your cancer B cells, but it doesn't work the same in all people. And at the end of the phase 2 trials, the drug Chimeric is showing to have 83% remission, which is really great for people who no other treatment has worked for, but there's still 17% that it fails for. So they're looking into different things. So like CD20 and CD22 are two other markers that are found on B cells that they might be able to target directly or in combination with CD19 to make it a more effective therapy for some of these patients. Additionally, like I said, combination therapy is a big thing they're looking at, not only using multiple different CAR T cells potentially, but also combining it with other drugs. The immunotherapy is not limited to just CAR therapies. There are other things that affect your immune system, such as your T cells, for instance. naturally in your body, they are programmed to become exhausted so that they don't come to a point where something goes wrong with them and they start attacking your own body cells. So to prevent autoimmune issues, your body exhausts these T cells. But cancer takes advantage of that. So now we're looking at ways to turn these T cells back on to invigorate them. So that could be used in combination with CAR therapies to get these T cells to be long-lasting and actually get rid of all remaining cancer. Because if there's just one cell, it's going to come back. An additional thing they're looking at is solid tumors are a big uphill battle for immunotherapies because there's a different sort of environment. With leukemias and lymphomas, you have this cancer that's floating all around your body, essentially. But with solid tumors, it's in a centralized location, and it's sort of built up this little like barrier. So the T cells can't even get in to get to their target with solid tumors. So now we're trying to find ways of essentially making an opening for these T cells. And just to wrap things up, this is my lab. We are called the Translational and Correlative Studies Lab. And our main function is to basically all the CAR T-cell trials at the University of Pennsylvania. These patients, when they're enrolled in a trial, they have standard time points that they have to give. Usually it's just blood, sometimes it's bone marrow, different samples come in. My lab receives all those samples, and then we do a variety of testing on that, but the basic thing that we need to tell clinicians is what the CAR therapy is doing. So is it actually expanding and proliferating as we hope it is? And what is the tumor doing? And hopefully the CAR is going up and the tumor is going down. And this is my group as a as a side. The woman on the far side in the red, she was actually the one that got the data to the doctors that saved Emily Whitehead's life. And she is the director of our biochemical section in my lab. All right, any questions?
Is that just a one-time process where you extract the plasma, do the thing, and then re-inject, and then it reproduces from there? Or do you have to extract more and keep replenishing?
In general, it's just once. They grow up enough where we have stores. So sometimes our patients, different trials explore different things, like reinfusing the patient. So they'll get whatever the initial protocol calls for. They'll get their initial 3 dosages of the CAR therapy. But then if they feel like they need a boost down the line, they might give them another series of CAR therapy treatments. So as long as they have enough in storage, they don't redo that. But occasionally they will run out, and then they'll have to do that whole process over again.
Once it's re-injected or infused, how long does it take to act on the cancer itself?
It depends on the CAR. If we're talking specifically about CAR-19, which is the big one that got approved, usually it's around day 7 that we see the peak of the most CAR cells in the patient, and then at least by day 28, if not earlier, we can no longer detect any sort of tumor, assuming it's been, it's a successful case. So it's very fast.
Amazing.
Yeah.
When you spoke of the potential for a person's cells to develop memory, is this sort of thing, is that possible after, say, the patient goes into remission, that the patient's own cells then are able to recognize these specific cancers?
Yeah, that's the hope, because the CAR T cells are their cells. They just have a secondary fancy receptor on them. They're still their cells, so the hope is that these cells do make that bridge into memory, so that if the cancer does come back, that there's a T cell there that still has that receptor, so it still remembers and could go right away and take out any cancer. It doesn't always work that way, but that's the general hope.
I have a question. Have you done anything against solid tumors clinically? Any trial, any clinical aspect? Not in vitro. Are you allowed to say?
Yeah. At this point, yeah, it hasn't been something big. Right now, the focus is really more on figuring out, before the clinical trials, actually figuring out how to make it work appropriately. There's been some dabbling into solid tumors, but it hasn't been any like big trials as of yet.
My question, I guess, is, let me rephrase. Most of your patients are leukemia patients, am I right?
Leukemia and lymphoma.
Lymphoma. Did you have you been approached by other cancer patients that they also seek therapies in order to to expand this technology therapy?
I mean, I haven't. I don't deal directly with any of the patients. I'm sure there have been people that have come to Penn because we're known for the curing cancer, and they might miss the point that it's only certain types of cancer at this point. But there are so many clinical trials open, a variety of immunotherapies, so not just CAR therapies, that I'm sure there is a trial open at Penn for solid tumors. Just not something I'm directly involved with as of yet.
What's the scalability of this kind of therapy? I mean, it looks like it would be in competition with the pharmaceutical industry. So how would this be something that would be scalable, that would essentially replace that at some point, possibly?
I'm sorry, I don't understand what you're saying.
Well, could this be mass produced, I guess?
Yeah, that is the goal. Because, I mean, right now, the drug company Novartis, who Penn partnered with, owns Kymriah. They own the therapy. So yes, because of right now, since I believe they've gotten it down to around $450,000 per patient for treatment. So yes, there is great efforts going into streamlining this and to making this a more cost-effective, more accessible therapy. But something they've done with Kymriah is that if you don't have remission in 3 months, then you don't have to pay. So if it doesn't work for you, someone eats the cost. I'm not sure. I guess Novartis.
Back to the potential translatability of this for other cancers. Is the concept applicable? I don't know much about the science of cancers, but the idea of creating a T cell that has the antigen to go after a specific cancer, it seems like other cancers might require different antigens, but that's a lot of work. It has to be invented yet, but is the concept at least applicable to other cancers?
Yeah, 100%. Right now, a big part of the field is trying to identify any sort of unique markers, because what you need is a marker that's not on a wide variety of cells, that it's only on the cancer or cells that you could live without. So that's the biggest problem. Fortunately, like I said earlier, a lot of cancers do kind of have a unique pattern that differs from normal cells. So they're trying to investigate that to see what separates the cancer cells from normal cells, to see if there's anything there that they could use. But for instance, there's a target called BCMA on multiple myeloma cancer, and they have a CAR now for that we're investigating in clinical trials that targets, and that's another B-cell cancer, but it's different. It's a later stage B-cell cancer, so it's different. The CAR-19 doesn't work as well with that cancer. So yeah, it's really the hurdle is just identifying. Yeah. So.
I'm sorry, I have to interrupt you, but the time does not allow to continue. I want to present, if you have more questions, you can ask her after this. I want to present Chelsea with a certificate of appreciation from STEM, and also a honorarium on behalf of the Pearson Education. So